Good afternoon, everybody. Thank you for joining us. My name is Ariel Handelman. I'm the Senior Director of Marketing and Audience Development, and welcome everybody, all of our wonderful members and patrons. Just a few housekeeping items we wanted to go through today. Throughout the presentation, if you have any questions, there is a Q&A button at the bottom of your screen. Please enter any questions that you may have at the bottom there, and we will share them with our presenter. If we are unable for any reason to get to any of your questions throughout, please do feel free to email them to membership@nybg.org, and we will send them to the presenter later. We'll also put that email in the chat function. Lastly, this presentation is being recorded and will be shared to all members and patrons via the member monthly update. Today, we are going to learn about the amazing William and Linda Steere Herbarium located at NYBG. It is one of the largest collections of preserved plant specimens documenting plant life around the globe over the past 300 years. We are joined today by the Assistant Director of the Herbarium, Nicole Tranowski. Nicole has worked at NYBG Herbarium for 24 years. Yes, she started when she was 5, and is also a Bronx native who grew up right around the corner on the Grand Concourse. She frequently visited the garden as a child and is going to share with us a virtual presentation of some of the popular specimens from the Steere Herbarium and some of the other wonderful, amazing things about this place. So take it away, Nicole.
Thanks, Ariel. Welcome. I'm really pleased to be here with you today and to have this opportunity to share some stories about the herbarium. For those of you that are not familiar with herbaria, we are going to start with a short video just to kind of introduce what our collection is all about.
Hello, and welcome to the Herbarium. I'm Nicole Tranowski, Assistant Director of the Herbarium, and I'm excited to give you a behind-the-scenes look inside the herbarium of the New York Botanical Garden. Our herbarium is the largest in the Western Hemisphere, holding approximately 7.8 million specimens, documenting the plants of the world over the last 300 years. The collection is housed in a state-of-the-art, climate-controlled facility within cabinets designed specifically for herbarium specimens. In fact, this design dates back to the collections of Carl Linnaeus, who created the system of taxonomy that scientists still use today to name and categorize natural history collections. The herbarium is curated with a meticulous filing system organized both by the scientific name, the family, genus, and species, as well as by the geography of where the plant was growing when it was collected and preserved. Each specimen consists of the physical plant, which has been pressed and dried, much the way one would press a flower in a book. There is also a label to tell you what the specimen is, its scientific name, exactly where it was collected, including latitude and longitude, which allows us to map it. The label tells you who collected it and when it was collected. This information is as important as the specimen itself. Preserving herbarium specimens has been done using the same process for close to 500 years. It is still the best way to preserve plants for study. The specimen is dried and pressed in the field using a traditional plant press. The specimen is arranged in absorbent paper such as newspaper, making sure to show all of the most important structures needed to identify it. It is sandwiched with corrugated cardboard, a wooden frame, and tightened with belts. Once the specimen is dry, it is sent to the herbarium where we can do the final step in the preservation process, mounting it onto a sheet of paper so that it is supported underneath and can be handled as it is studied. Our mounters will first arrange it on the sheet, being sure the important structures are visible, but also with an eye for design. Obviously, these specimens are created for scientific use, but there is also a beautiful art to preserving and presenting the specimens in this manner. Every specimen gets a small packet so that we can save any pieces that may fall off. These fragments are, in fact, very useful to researchers as they can extract DNA from them. Recently, one of our specimens, originally collected in 1829, was sampled and DNA was able to be extracted. We then drizzle glue on the back of the specimen and attach it to the paper. The glue is a water-based white glue that is similar to Elmer's, and it is acid-free and archival quality. While the glue dries, we weight down the specimens and cover with wax paper. To reinforce gluing them to the sheets, many specimens are also secured by sewing them to the paper. In contrast to the traditional practice of mounting herbarium specimens, these days we also focus our efforts on digitizing our specimens so that we can share them online with both researchers and the public. Our digital imaging lab contains equipment specifically designed for herbarium specimens. We use high-resolution instant capture cameras and light boxes, which allow us to provide diffuse lighting for each specimen image without needing to use flashes. The image is taken and checked on screen. You can see the level of detail in our images by zooming in to see the many important structures of a specimen. The herbarium contains many treasures and its contents tell many stories. I'm looking forward to telling you more.
Great. Well, I hope you enjoyed that video and got a little bit of a background on kind of the journey these specimens take from the field to come to live in the collections that we have here at the garden. As I mentioned in the video, these specimens can tell many different stories, and I thought we would start with some of the earliest known herbaria. The earliest known was created by Luca Guinea. He was a physician in Bologna, Italy in the 16th century. And these very early collections were pressed and dried and attached to paper almost identically the same process that we do now. The reason for these collections was a little bit different. They were documenting medicinal plants and sort of creating a reference collection. So these herbaria were all bound sort of in books. And so this really was sort of the beginning of the European tradition of scholarly studying plants. So Luca Guinea and then many of his students started to generate these herbaria and these collections. And for the next century and a half, this spread throughout Europe as a practice for studying these plants. By the 18th century, Carl Linnaeus had become a botanist, studying not just plants, but also all different types of natural history. And he was really the first botanist to advocate mounting each specimen on its own separate sheet of paper. And there was a good reason for that, his research had sort of shifted from only studying these as medicinal plants to studying the plants themselves and being able to document them and categorize them. So to put every specimen on its own sheet of paper meant that you could then shuffle your ideas about what plants might be related to other plants. You can revise your ideas. You can pull that one individual plant out of the collection and maybe file it somewhere else that made more sense, that maybe it was more aligned with. So you can see this image on the right is actually an illustration of Linnaeus's original herbarium cabinets where he stored his own herbarium. And so he advocated in one of his publications, Philosophia Botanica, that all botanists should start storing their specimens in this manner so that it became easier to categorize them. Some other early collections that tell some pretty neat histories are collections that we house at the garden here. Some of our oldest collections are from the Captain Cook expeditions. So Captain Cook's first expedition, this was a British expedition, right? This was sort of in the age of exploration and also colonialization. So Cook was sent by the British Empire to travel the whole world, and brought along a whole host of different people to document what they found out in the world, including two botanists, Joseph Banks and Daniel Solander. So every stop the ship made, Banks and Solander would get off the ship and make collections. This is an example of a specimen we hold here at the New Botanical Garden. This was collected on that first expedition, so probably 1770, 1771, in New Zealand. And if you can see on the label here, it says type specimen. That represents a specimen that was documenting a new species that Western science was describing for the first time. Obviously, the cultures in New Zealand already were familiar with this plant, and so this was sort of formally in this Western tradition of studying botany, our way of categorizing these plants. And we still document brand new species in the same way. A researcher might look at 200 specimens of a particular species before they really decide that it's a brand new species, but they always choose one to represent what the characters of that species mean. Other illustrious botanists represented in our collection include Charles Darwin. This is a specimen that was collected in Tierra del Fuego on his voyage of the Beagle. And you'll see, you can see how many different types of labels and annotations are on this specimen. I've blown that up for you a little bit here. And this really represents how we continue to handle these specimens now. The entire journey of a specimen, as much as the botanists themselves are journeying all over the world to collect these specimens, once they arrive in the herbarium, the specimen itself sort of continues its own journey. And anything that has happened with that specimen, including any botanist that has studied it, will record information on that specimen about what they used it for. So in this example, you can see there's all of these illustrations. This is a little moss collection that Charles Darwin made. And these illustrations were actually made by another botanist named William Mitten. So much like today, even back then, the person that collected the specimen was not necessarily the person that knew what it was. So at the time, William Mitten was the bryologist, the specialist in studying mosses. And so Darwin sent this collection to Mitten to be identified. And Mitten was able to keep it for his own collection. And we still do the same thing even today. You can see there's all of these other stamps. Again, there's one that says type. So again, this is a type collection. So it represents a brand new species that Darwin collected and that Mitten named as a brand new species. You can see there's all kinds of other labels on here. It was apparently loaned for an illustration at one point. So it sort of tells, the entire history of every individual specimen on its sheet. Other fun historical stories that our specimens can represent sometimes are tied to the history of our own garden. If any of you are familiar with Elgin Garden, this was considered the first public botanic garden in the United States. And it was where the footprint of Rockefeller Center is today. There's still a little plaque there. If you make it to Rockefeller Center, you can see a little plaque that said this used to be Elgin Garden. So this was established by Dr. David Hosack, who was a practicing physician. He's actually also famous for being the attending physician during the Hamilton-Burr duel. He had been friends with both families and was the attending physician that day. But in addition, he was also an avid botanist. Much like those early traditions of Luca Guinea studying botany for medicinal purposes, every physician had to have a pretty decent education in botany, and that really piqued David Hosack's interest in botany. And so with his own funds, he established this garden in New York. It contained 2 greenhouses, and he had a herbarium. And he was able to keep this going for a number of years. After a while, it became really expensive for him to continue, and he ended up ceding the property to the city of New York. Yeah, so by 1816, I think this had been ceded to the city, who then in turn gave this property to Columbia College. So you can see on this specimen, so all of those herbarium specimens became part of Columbia College. And again, you can see the history of some of this, on this specimen. It says that it was received from Elgin Garden in New York in 1816. And then later it became the property of the herbarium of Columbia University. And so that continues to tie into the history of our garden. So this was in the early days of Columbia College, when it was on the property of Columbia College. This was their 49th Street campus. And one of the most prominent botanists at the time in the entire country was John Tory. He was a botany professor at Columbia. He was well known throughout the country and the world. Most botanists throughout our entire country were collecting specimens, sending him duplicates. So he amassed his own personal collection of over 50,000 specimens throughout his career. And towards the end of his career, as he retired, he donated the entire collection to Columbia University, and he started the Tory Botanical Society. And so this was a society he really created to allow both botany students and amateur botanists to get together, go on field trips, and talk all things botany. This is a really wonderful picture from our own Mertz Library digital collection of the Tory Botanical Club taking a field trip up here to the Bronx, getting off at Pelham Station in 1893. And so that sort of continued the history of botany in New York. And that brings us to these two illustrious young botanists at the time. Nathaniel Lord Britton and his wife Elizabeth Britton were the founders of the New York Botanical Garden. Nathaniel Lord Britton was actually a botany student at Columbia. He enrolled just 2 years after Tory died. I don't know if they ever actually met. But in both of their young budding botany careers, they became part of the Tory Botanical Society. They were very active members, went on all the field trips. And these are some examples of specimens that the pair of them collected together. This specimen is especially sweet to me. It was collected in 1883 before they were married. So they got married in 1885. This was a collection made up here in the Bronx in Kingsbridge. So I sort of like to think this was, sort of part of their courtship was going on these botanical club expeditions. And on one of these journeys up here to the Bronx, and sort of foreseeing maybe where the garden was going to land, Nathaniel remarked on visiting the Bronx, "It was this trip that gave me my first knowledge of the natural beauties of the Bronx Valley." So the Brittons were married. They, for their honeymoon, went to London and visited Kew Gardens, which had already been in existence for 100 years. And they were just incredibly impressed with the institution, really as a scientific research institution. And upon returning here, they really felt strongly that the United States needed as equally an impressive research institution for the study of botany. Elizabeth in particular, when they returned, addressed the Tory Botanical Club on just October 24th in 1888 to establish a botanical garden. And so the club published this public appeal, to gain support for this. And by 1891 is when the New York Botanical Garden was first incorporated. And so the first board of managers included Andrew Carnegie, J.P. Morgan, Cornelius Vanderbilt, and Nathaniel Lord Britton. This is another great photo on the right here, again from our Mertz Library digital collections. So this is the Tory Botanical Club taking possession of this property for the very first time. So the hillside that they're walking up I'm sure many of you are familiar with our own Tulip Tree LA. So this is actually that same hillside that they're walking up before there were any buildings, before there were any tulip trees. This was the very first time they had set foot on the property. And we continue these traditions, you know. So when the Tory Botanical Club first established the garden here, they took the entire herbarium of Columbia University. They brought that up to the garden here. That was sort of the nucleus of our initial herbarium. And since then, every leadership role we've had at the garden has continued to make sure that our collection has grown over the years into what is now one of the largest collections in the world. We have 7.8 million specimens. And every one of those specimens tells you an important piece of information. Every single specimen is representing that particular species collected in that particular place at that particular time. And so each one of those pieces of information can be used to study plants. You know, one of the first things one needs to do, even if you're an amateur botanist, if you've ever used a field guide, a wildflower guide, is, you know, you might be walking around saying, oh, what is that plant? I want to know what it is. And that's exactly what botanists are doing as well. So, you know, someone might use a wildflower guide, which might be organized by color, flower color to start with. Scientists often use what we call these binomial keys, right? So they would sort of lead you through a set of questions, and for each answer to that question, it will lead you to another question, sort of like a choose your own adventure, but it sort of leads you to discovering exactly what that species is within that key. The other important use of our herbarium in terms of trying to figure out a plant that you may have collected is using the collections we already have here, right? So one of the biggest strengths of a herbarium is that every specimen in this building has been collected in a different part of the world, is a different type of plant, and has been collected over time. But one of the strengths is that they're all in the same place. So we're really gaining from all of the previous work that previous botanists have done over the last 300 to 400 years. And we can use what has already been done to help us as a reference collection to figure out what we're currently working on. So that's one way that botanists can focus their type of study. They might be a specialist in a particular plant group, right? So this, for example, is the Ericaceae, the blueberry family. And so they might be studying everything just about that family in terms of how the different species within that group are related to each other, how they're related to other plant groups, and sort of drawing a family tree to see how plants have evolved over the years. It also might include species concepts, really detailed descriptions of each species, beautiful illustrations really meant to describe, you know, in a visual way, just accompanying descriptions. Another important piece of information on the specimens, of course, is the geography. And again, because we might have so many specimens just of one particular species, if you start to plot all of those on a map, you can really get a sense of the entire distribution of where a particular species occurs, right? So if we have, this is the Brazil nut, this is a species of Brazil nut tree, Bertholletia excelsa. And we have hundreds of specimens of this that have been collected in many different parts of North and South America. And you can see each one of these red dots on the map represents a particular specimen, right? So in mapping them in this way, we can start to see, you know, the northern reaches of where the this species can occur, the southern, eastern, western reaches of where it occur. We can start to layer those maps, so we can start to figure out, does it grow near water? Does it grow in drier areas? So that mapping capability becomes really important in terms of studying the geography of that particular plant. Also, in knowing the geography of a particular plant, you can, instead of studying a particular plant group, one could study a geographical region and study all the different species that grow in that one geographical region. So this, for example, is a publication called Intermountain Flora. Two of our researchers, Pat and Noel Holmgren, have spent a 50-year career documenting every single species that grows in this region of the Southwest US, basically everything between the Rocky Mountains in Colorado and the Sierra Mountains in California. Another aspect on these specimens, because each one of them has a date, is that you can map a species over time. And one, especially with climate change research, we can start to see how plants have reacted to a changing environment. So herbarium specimens have increasingly become part of phenological studies. So a phenology is the study of at what time a plant is developing, right? So in this case, most of our herbarium specimens, almost every herbarium specimen, when it's collected, they always try to collect either fruits and or flowers, because those are usually some of the most important characters needed to identify it. They've also become really important in terms of studying phenology, right? So you can study a particular species. These are all exactly the same species of viburnum, collected in almost the identical place, but you can see there's this other dimension of time. And we can see how the flowering times have changed. This first specimen was collected in 1891, and it was flowering in the third week of June. Much later in 1972, another viburnum again was collected in exactly the same place, and it was flowering earlier as springs become warmer. This last specimen was collected in 2011 and was actually flowering weeks earlier in the fourth week of May. And obviously these are only an example. Most phenology studies use a lot more statistics and many points of data, so that there's a statistical analysis that goes along with this. But it really shows you how these preserved specimens can help to document how the plants are reacting to their changing environment. Another thing that can be studied in relation to time is invasive species. This is an example of purple loosestrife. Some of you might be familiar with purple loosestrife. It's a very pretty, terrible weed that was introduced into North America from Europe. This particular herbarium specimen that we have here in the garden is considered one of the earliest collections documenting that purple loosestrife had established itself here in North America. So you can see on the label here, this was collected in September of 1831 in New Bedford, Massachusetts. And of course, there have been, many herbarium specimens collected of that since that time. And so we can really track every one of those specimens on a map and really start to see how it spread throughout the country. This is another study that was done on purple loosestrife in the Great Lakes region. You can see how there was just one collection made of it in 1883. By 1950, it had really spread throughout the Great Lakes. By 1975, you can see how it's growing and growing even more. So it's an interesting use of herbarium specimens, again, as data points to really see where plants are moving. Plants are constantly moving and reacting to changing environments. Herbarium specimens can also be rare collections. This is a really pretty specimen of a Trillium, a very rare species that grows in Georgia. And there can be many reasons for a plant to be rare. It can be often, as in the case of this Trillium, They can be endemics, which means that they are a species that really have a very tiny range in terms of where they grow, right? And so those endemics are often much more susceptible to changes in the environment, to habitat loss, and that kind of thing. So this is a trillium that is very rare because it started off as an endemic, but it's also had habitat loss and changing environments can affect that as well. We sadly also have many extinct species documented in our herbarium. So this is a specimen that was collected in 1910 in Hawaii. It's a pretty neat looking specimen. It's in the aster family, in the daisy family. And again, Hawaii is the perfect example of something that is, this very tiny, region, island in the middle of that where there isn't any other land for the species to go. So as there has been habitat loss and as there's been climate change, the places that these plants can actually continue to grow and survive just keeps shrinking and shrinking. So unfortunately, we do have some specimens in our collection that represent species that no longer grow on Earth. I should point out that the majority of collections that we have in our herbarium are plants that are growing in nature. They're not cultivated. We're traditionally, we're not a seed bank. We're not, the point of our collection is not to have viable seeds that could be used at a later point for germinating. It's really supposed to be here as a reference collection. But in saying that, we do have collections of many wild relatives of cultivated specimens. So these are so this specimen on the left is actually domesticated corn. And the specimen on the right is Teosinte. So this is a wild relative of maize that naturally occurs in Mexico and parts of Central America. And it's very interesting to think about studying wild relatives of crops, especially as climates change and it becomes more difficult to cultivate some of our crops, whether there is less water or higher temperatures. Often some of these wild relatives can be studied for characters that might be able to survive these more drastic and extreme climates. And so we might be able to use some of those characters to introduce back into our crops in order for our crops to survive, more heartily in changing environments. Molecular biology has also been a huge part of studying botany in general in the last 30 years. Information taken from herbarium specimens and sequenced from all different types of plants has really helped us in terms of figuring out relationships between how different plant groups have evolved, and sort of rearranging some of those family trees in being able to use this molecular data. There's been other interesting uses of this molecular data more recently as well. Again, sort of in terms of thinking about climate change and changing environments. Most recently, we had a group of researchers that came to visit from the University of Florida, and they were doing a really interesting project where they sampled many different species from our collection, and they were studying how different species use nitrogen fixation, which means so there's some species, many in the legume family, that can actually grab nitrogen on their own without needing to absorb it from the soil. And so the idea behind this was that if they can sort of isolate some of those genes that are involved in nitrogen fixation, maybe we can use them for other crops that aren't able to use nitrogen fixation on their own. But if they could in the future, maybe it could cut down on using being able to add to the soil. So those are some different aspects of scientific uses of specimens, but of course, as you could see even in the video, these specimens really can be considered works of art themselves. Every specimen, because each one is put on its own sheet of paper, are really like a portrait. And even the process of gluing them down and sewing them is sort of this interesting craft that is really only taught within the world of herbaria. It's not really something one would really learn anywhere else. And so even within our own institution, our own sort of tricks of the trade have just been passed down through generations of our own mounters. So they're really, you know, as important as these specimens are scientifically, many of them are very beautiful and can be considered art themselves. And the herbarium in general can really be appreciated as a museum. It really is an archive of these plants. You know, every single one of these specimens, once it's pressed and dried, it's basically preserved for centuries. It'll be cared for in our collection and in our state-of-the-art collection here with our climate control and cabinets that keep the specimens safe from mold and insects that could possibly damage them. This is another great image up on the left from our March Digital Library of what our museum building looked like when the garden was first opened. That main floor was a museum collection, showing a lot of ethnobotanical collections of, you know, how different people can use plants. And so I love some of these old, you know, sort of Victorian looking curio cabinets to really show off some of our specimens. And so we continue today, still continuing to care for the collection, hoping, you know, we're stewards of this collection that we hope will continue to be here for researchers for centuries to come. And with that, I thank you. And I guess we can open that up to some questions.
Hi, Nicole, and thank you everybody for bearing with us. Sorry, we were having some technical difficulties with this, these black boxes that are showing up on the screen versus the transcripting. We're not really sure what exactly is going on, but hopefully everybody was able to hear, and we are recording this. This will go on the website. So if you missed anything, it will be transcribed and up on the website later. But Nicole, we have some great questions for you. One of the first questions that came through, is there an opportunity for volunteer service at the herbarium while working from home?
Yes, there absolutely is. I didn't talk too much about our digitization process, but you may have seen in the video that we are currently digitizing every one of our specimens in the collection. We've actually been doing this for 25 years. We're just about at the halfway mark, but we've sort of sped up over the years. And you know, we hope to actually finish digitizing everything within the next 6 to 7 years. But what we do these days is we take an image of every specimen, and then we have been using crowdsourcing to help us go back and look at the label information on every one of those specimens so that we can put all of that data into our database. So we have some links that we can share with you for our herbarium citizen science projects. We have several, we call them expeditions. We'll sort of group specimens so that people can, you know, choose a particular expedition and look at the particular specimens in that group and help us out to transcribe the specimen records.
Great. So we'll add some of those links. So then another question that came through about the purpose of herbaria to collect, and I think you answered this in various ways, but just to kind of really kind of push the purpose and stuff home about Is the purpose of the herbaria simply to collect samples for research and study? Given the size and longevity of the collection, does NYBG map the decline of botanical species in various locations? Conversely, given the acceleration of global travel and effects on climate change, has NYBG been able to track the expansion of various botanical species? And yeah.
Yeah, absolutely. That's become, you know, a really important use of our specimens because we have this record of where particular species were growing on Earth over this long period of time. We can absolutely track how different specimens are moving. So that includes not just invasives, but even native plants that maybe, you know, even within the U.S., there are species that used to grow much more in a southern climate that are slowly moving north as the climate gets warmer. You know, obviously invasives are plants that kind of travel with people often. So yeah, this has been, because we have that geography information on every single specimen, it's really useful information to help us track the specimens.
So there was a chart that you had referenced a lot early on. Someone was asking about the color chart early on, and I don't remember which page that might have been on. It was.
I can go back. Yeah, so all of our specimens have a color chart added to them. Let's see, I can find one. This is one. Yeah, so the Captain Cook specimen. So this has been, because we've been digitizing our collections for so many years, we kind of came up with a lot of the standard ways of doing this for the entire herbarium community. So you'll see actually that every single one of our specimens has a color card like this so that you can standardize what the true colors are on the specimen. So these color cards are sort of standard colors, and you can standardize how that's seen on your screen. You'll also notice every specimen has a ruler so that someone could actually, you know, if a researcher needed to measure things on a particular species, they have that ruler to standardize a way of measuring on the specimen as well.
So, and I think that another question just came in about how do you preserve the colors, especially in flowers? And I think that the colors has always been a very important aspect of what you guys work on. You want
Yeah,
to kind of elaborate a little bit about that with both the color chart and.
yeah. I mean, there are some plants that we're just not going to preserve the colors, but you know, especially things that have been collected in the tropics. You know, you think of these lush, beautiful tropical flowers, and often herbarium specimens made from tropical plants because they're in such a wet environment to begin with, and the plants themselves have so much water in them, they all turn brown. But it really depends on the particular species whether they're going to hold the color pretty well or not. So traditionally, herbarium specimens don't necessarily rely on the color of the flowers, but there are often plant descriptions on the label that give you a sense of what the colors were before the entire plant was pressed and dried. And also the descriptions that botanists make when they're describing a brand new species, for example, they have to make a really detailed description of that species, including colors and all of the measurements. So you can't really rely on the colors in the herbarium specimens to be true to nature anymore, but there are sort of other tools that botanists include when they're thinking about color.
Someone was also asking about do you also document the types of insects, birds who thrive on these plants? And then I guess another question is also about how then climate change has affected them, about plants blooming earlier than ever or different blooming cycles.
Yeah, so those are two different. So yeah, sometimes a collector will include something about the insects they found living on a specimen before they made it into a herbarium specimen. Occasionally, our mounters will find some little hitchhikers, you know, poor little insects that got pressed in the plant press with the plant specimen, so they were kind of preserved I think we even found a little tiny frog on a specimen once. But primarily a herbarium specimen is kind of considered a portrait, as opposed to a landscape that's showing everything in a picture. A herbarium specimen really is supposed to, is just highlighting and isolating just that one thing. So really herbarium specimens individually are not considering their entire the entire ecology or habitat around them. But they certainly can contribute to the data of say an entomologist that's working in the same region that might, if they have the same sort of geographical information, that data can be worked together. And to answer your question about climate change, yeah, certainly there are phenological studies, obviously on plants flowering earlier, and of course that's going to affect pollinators, right? So if a certain type of bee is specific to a certain type of flower, and the flower flowers 2 weeks earlier than it used to, and the bee is just hatching when it always does, they're going to miss each other, and that bee might miss a chance to collect pollen for food, and the flower might miss that chance to be pollinated. So that is a problem.
Someone asked, which I think is always helpful, if you could explain phenology again.
Yeah, absolutely. So it's the study of sort of how an organism develops through time, right? So it's, if you think about a tree that, if it's a deciduous tree, it lost all of its leaves, it was, over the winter. And then in the spring, you start off with little buds, and then at some point those buds burst into full bloom, and then maybe later in the season they start to develop fruit. So it's that development that we're studying over time. And that's the study of phenology.
Cool. Is the herbarium open to any member? How can one visit it? Which is exactly why we're doing this.
We happily do these virtual tours during pre-pandemic times, and hopefully post-pandemic times. We do several open houses a year. Normally the herbarium is not open to the public. It's available by appointment, mostly for researchers, or really for anyone that has a legitimate academic reason for needing the herbarium. We've had many historians and artists come to look at the herbarium and consult specific specimens. So most of the year is not open to the public, but we do like to host these open houses where 2 or 3 times a year we'll allow the public in to look at our specimens, and we have a lot of staff on hand to interact and answer many of the questions. I should also mention we started an online version of our herbarium that we call the Virtual Herbarium, and a portion of that we have created what we call the hand lens, which tells many of these herbarium stories, and that is open, available to anyone that would like to learn more about the herbarium and many stories that we can tell them.
And this is, again, why we love to do these things for our wonderful members and patrons, which we thank very much. Sorry, have to. So another question has come through. Has anyone been able to determine how many species have gone extinct by referring to world herbaria?
There is actually an international group that tracks species extinction. And that the extinct specimen that I showed you was taken from that list. It's called the IUCN Red List. So it's a list of every plant species that is thought to be extinct. And that is, that does use herbarium information in terms of, geography. If a botanist is trying to figure out if a plant is still in existence, they can use the geography on our herbarium specimens to go looking for it. And if they can't find it, they might determine that it's extinct. That particular specimen that I mentioned, we used the IUCN Red List, and one of our interns last year combed through our entire herbarium looking for extinct species. And so she found she found about 55 specimens that all represent species that are extinct.
I was just posting the blog post that was done. I think it was part of it, the plants that had gone extinct in the past 250 years that was related to that. So that was a great question, and I remember us talking about that when that happened. So somebody asked, recently heard that we are losing a significant amount of global wildlife due to the warming of our planet. Is there an acceleration of decline in the botanical world that you could elaborate on?
Yeah, sadly, that the botanical world is also declining. I mean, we continue, botanists around the world continue to go out and study and collect specimens on going on field trips and documenting in many different places in the world. Some of our own researchers are working in regions that are quickly disappearing. Doug Daly, one of our researchers, has spent his entire career working in Amazonian Brazil. If you remember the horrendous fires that happened last summer, he went out to the state of Rondônia on some field trips to go and document many different, the loss, the incredible amount of habitat loss. And he was also there in search of a few different species, some of which he found, some of which he didn't. So yeah, our own staff is very much involved in documenting the loss of species, of plant species.
So there's a couple questions, and I think this all kind of relates to about the collection of our own specimens, as well as somebody asked about our collection still donated from universities' private collections, and was asking about our recent acquisitions. And it just made me think especially about the project with the Rondônia project, about species being sent in. So maybe you could share a little bit about that process, and even about the amazing work. I'm sorry, I'm boasting about how amazing the team is, but just the amazing work that you guys all did during even COVID, and pieces being sent, and the work, the species that were being sent even from Vanuatu that were sent. If you could just, I think the audience would really get that's a wonderful story about how we worked and how your team really worked through the pandemic of collecting specimens then.
Yeah, absolutely. Yeah, that's a great question. I didn't elaborate too much about how we acquire specimens, but every researcher that goes out into the field, when they collect plants, they'll actually collect duplicates of each plants, right? So, for that Brazil nut tree, for example, when that was collected, they probably took down a whole big branch and were able to clip it up into probably 7 or 8 different pieces, each of which were from that same individual and are considered that same collection, but they're duplicates of each other. So that allows us to keep one for us in New York. It also allows us to leave, and this is very important, to leave specimens in the country, the host country, where that plant was growing, and where they also have important herbarium researchers. So we always need to leave material in the original country. And then we usually have additional duplicates to share with other herbaria. So that's a way that many herbaria are helping each other to grow. We're constantly trading specimens with each other. So if we have extra specimens, we're going to send some to Kew or to the Smithsonian, and they're going to turn around and do the same for us. So we're benefiting from their collectors going out into the world, and they're benefiting from ours. And to talk about the Vanuatu project, one of our researchers, Greg Plunkett, has been working in this, Pacific Islands of Micronesia and Vanuatu for many years now. It's a fascinating project. It's a group of islands where there really hadn't been a lot of documentation, at least Western documentation of the plants that grow in that region. And because of habitat loss and climate change and cultural change, there's been a lot that the people that are from that region have had a renewed interest in documenting those plants themselves. They also, there's something like well over 100 different dialects spoken in those islands. So it's been a really fascinating project in a lot of different ways, to document not just the plants, but the culture surrounding those plants and the interest that they have themselves of preserving their culture. And so Greg was in Vanuatu when the entire shutdown happened. And this is a tiny little place. There were no flights in and out of Vanuatu for months. But he finally managed to, and he had been collecting specimens for a long time. He had hundreds and hundreds of specimens. He was working in the small herbarium they had set up there. But he had to find a way to get those specimens, the duplicates destined for us here at New York. He had to find a way to get them shipped to us. So it took a couple of months. I think they had to maybe be put on a boat before they got to somewhere else that was put on a plane to get here. But they took quite a journey. The specimens themselves took quite a journey just to get here. And we were here waiting for them when the specimens finally showed up. They had to go through the USDA and all of our regular permitting process. But they arrived safe and sound, and we now have those specimens in our herbarium here.
So somebody asked a question about, does the herbarium also contain live herbarium plants? And I'm just thinking of the process and of the freezer that goes through. So you want to?
Yeah, so we don't have any live plants. So our living collections here at the garden are in the conservatory and out on the grounds, but the herbarium is all dried and pressed specimens. They're not living. We do, Ariel mentioned, we do freeze all of our specimens, but that's primarily to kill any pests that may have come in with the specimens. I didn't mention too much about the archival aspect of how we care for our collections, but the reason that we put these specimens in these metal cabinets is to protect them from beetles that love to eat dried plants. There's a dermestid beetle called a cigarette beetle that can completely decimate a herbarium collection and turn the entire thing into dust. So we have a pretty strict pest management system to prevent insects from even entering our collection. So we freeze all of our specimens before they're brought into the herbarium.
So there was a question, one was about how many thousands of species are each North and South are in each North and South American country and continent? Do you know offhand?
I don't know offhand. That's a great question. The number of species worldwide is estimated to be between 300,000 and 350,000 worldwide. There is a much greater diversity in the tropics. So the regions closer to the equator tend to have much higher diversity. So there's going to be a higher number of species closer to the equator than there are in temperate regions like, North America and the very bottom of South America.
And so then an earlier question had asked about then, given the loss of herbaria such as Manila and Berlin, are our specimens segregated from the general collections?
We do separate our type collections from the rest of so the herbarium in general we're in our own building, our own separate building from anything else at the garden. And the entire thing is climate controlled. We do within that collection, and you're right, because during World War II, many herbarium collections were lost in Berlin as it was bombed, including many important type specimens. So this one that I left on the screen here, I mentioned this is a type specimen of a species. So that was the very first specimen upon which that very first description of it was made when it was first became a species, as in this Western scientific understanding. So our type collection are considered our most important collections. And we do keep those in a separate part of the herbarium than we do from the rest of our herbarium specimens. When I mentioned how we collect specimens in duplicate, that's sort of another insurance policy on damage that could possibly occur in any herbarium. You know, since we have these duplicates, we've actually spread that collection out to 7 or 8 different institutions so that, God forbid, if something did happen in one collection, hopefully there are duplicates in another institution that can carry on that representation of that particular specimen.
Thank you, Nicole. It's 4:03 and this has been fantastic. And thank you guys so much. There's even more questions that we haven't been fully able to get through. So we'll definitely pull together some of these questions and try to get some more answers from Nicole to share. But I highly encourage everybody to check out our read and watch section on the website, because we bug Nicole a lot for things on our blog post, and we do have some great topics, as well as to look at the hand lens section in the plant research and conservation section of the website, where there's a lot of other wonderful bits and bobs of information, as well as videos and some other topics on some of the projects that Nicole has talked about. We will have this video fully transcribed and recorded and up on the website soon in our lecture library, which is in the Learn section of the website. So you will be able to rewatch this and see if you missed anything. And we thank you, Nicole, tremendously for your time and your work and your dedication to NYBG. We thank our members and our patrons for their support. And thank you, everybody, for spending the afternoon with us. Happy Friday. And yeah, hope to see you guys at one of our next events.